know a lot of people are kind of relying on it.
Would you be able to tell me how many people are actually online at any given point in time?
Yep, I should be able to. Okay, this is what I'm seeing. Is that good? That's good. Good for me. At these points, what else you can do? I should see if it's playing. Yeah. Okay, we're good. Well, I'm gonna steal my cookies. Thank you. Thank you. Thank you.
There's still lots left. There still are lots of baked goods left, so please help yourself. So there's still some muffins as well as some cookies. Okay, so just wanted to go through really quickly. If you are writing the upcoming makeup test, please make sure that you're on time. There are only 4 questions. There are some parts associated with it. It should not take you the full 60 minutes, but it is a different format. So just to let you know that it is a different format. Again, it's on Friday at 3 p.m. It's over in Wilson Hall, so you'll go to New College, but you should have all those details emailed to you from the HMB office. Okay, so we're going to go back and re-examine some of the different things that I started off talking to you about. I did not get to things related to mild traumatic brain injury nor about diffuse axonal injury, and I also wanted to give you this idea or this notion, very, very interesting notion that perhaps multiple sclerosis is caused by concussions. Again, it's a very out there theory. There have been no follow-up studies, but I wanted to go through some of their data in today's lecture as well. Also, I'm always interested in putting up a picture of Tom Brady because, hey, he's a really good quarterback and he happens to be the Super Bowl champion. So let's go back and think a little bit about some of our ideas about myelination. And when we are thinking about myelination and the importance of myelination, one of the reasons why we're looking at that is because myelin helps within the mammalian nervous system. It helps to speed up the conduction velocity that we can measure using the space constant, and that's primarily through that RM value that we looked at earlier. So one of the models that I indicated for you earlier, the one that's probably the most prevalent within research papers that you might read, is what is known as the EAE model for multiple sclerosis or for demyelination, and they're used interchangeably. And this is what the EAE model looks like if you happen to read a paper on it. So usually we start by immunizing a rat or a mouse. Usually in this case, what you're seeing is a rat. And that's because they're larger, they're easier to work with, it's easier to get the T lymphocytes out. And one of the things that we do is we take an immunogen, something that we want to raise an immune response to, and we looked at the different peripheral versus central myelin proteins. One of these is myelin basic protein, found both in CNS and PNS myelin. And we can use what is known as CFA, or complete Freund's adjuvant, which helps to deliver and further immunize the mice. It's just a carrier molecule. So this is the immunogen itself, and we can then combine it with CFA, immunize this rat, and this rat will develop an immune response that we are able to measure. And this rat here, after injection, may end up having paralysis. It depends on how severe the particular immune response is, but it becomes paralyzed primarily because the nerves start to degenerate as a result of this immune response that gets activated. So we can remove parts of the immune system that are easily identifiable, like the lymph nodes, and I'll show you the lymph nodes again on a different diagram. And this rat, which has this immune response, when we remove the lymph node, isolate the cells from the lymph node, and then add MBP, the same immunogen again, it creates an even stronger response, or the immune cells respond even more specifically. And so you will develop these T cells, and again, T cells are part of that immune response, that are then specific for myelin basic protein. We then will inject these T cells into a normal rat, and this rat will develop that experimental autoimmune encephalomyelitis, and this will resemble something that looks like multiple sclerosis. So this is one of the models, the most common model for things like MS. We know that it's a very good model because this rat here, the EAE rat, will have excessive inflammation. If we were to draw some of its blood, we would see that it has many of the inflammatory markers. In addition to that, there will also be induration, which is a hardening. So just like we talk about sclerosis, and sclerosis means hardening. So when you think about atherosclerosis, arteriosclerosis, it means your blood vessels are getting harder. Multiple sclerosis means that multiple systems within the nervous system are much more stiff or harder than they used to be. Induration is another medical term for hardening, which again is observed within the nervous system. So all of the hallmark features of multiple sclerosis are observed in this particular laboratory model. And again, we will talk a little bit about necrosis later on today. So this model, the EAE model, is one of the best studied. It does involve an autoimmune-like phenomenon. And we do get the inflammation in both the spinal cord as well as the brain. Not surprising, because when we look at where the immunogen is coming from, we have this myelin basic protein, which we find both in CNS as well as in PNS myelin. And it does involve these T cells, which I had alluded to earlier. The other form of EAE that is often introduced, if you're not using MBP as the immunogen, you might use something like PLP instead. And I had mentioned proteolipid protein very briefly in the last lecture as well. So eventually, this leads to demyelination, centrally, peripherally, in a number of places. It leads to things that look behaviorally like multiple sclerosis. And it also models some of the immune response, the inflammatory changes that we might see. And this then becomes a really good model system. That is one model system. On that sheet that you had that you were filling in last Monday, one of the other things that was asked of you was to make sure that you knew some of the other models. On here, there are actually 3 other models, and I wanted you to be clear on those. We can also, instead of developing an immune response directly, we can indirectly cause an immune response in 2 different ways. And I'll kind of separate these 2 things out. One of these, which is fairly easy to do, is known as the Cooper-Zone model. And I went through it very quickly. I'm going to take my time and go through it again today. Cooper-Zone is an agent that we can feed to rats. That's the most common way that we can do this, to either rats or mice. But again, I'll talk about rats being a little bit more resistant to demyelination using things like Cooper-Zone, ethidium bromide, etc. So this is more often seen in mice. So we will give Cooper-Zone, an agent, to these mice. And they ingest it, they drink it, or they'll eat it, or they'll do combinations of that. Cooper-Zone depletes the levels of circulating copper. So why is that important? Because one of the antioxidants that we find throughout the different cells within your body is known as copper-zinc superoxide dismutase, or SOD1. So without copper, this superoxide dismutase actually fails to function. It doesn't work well because copper is necessary for its enzymatic activity. And so as a result, it has this impaired homeostasis. There is no copper zinc superoxide dismutase, and particularly within a cell type that I didn't go through in a lot of detail, which are known as tanocytes. For those of you who are taking other histology courses or are taking neuroanatomy, these tanocytes are a form of ependymal cell. And these ependymal cells are the cells that line the ventricles. And these cells that line the ventricles are particularly found in areas like the third ventricle as well as the fourth ventricle. That detail really is not important. But just keeping in mind that these tanocytes are the ependymal cells or approximations of the ependymal cells that we would normally see within the brain. Again, why is that important? Well, we have, in these tanocytes, we would have copper zinc superoxide dismutase, or SOD1. And normally, these tanocytes are helping to maintain the cerebral spinal fluid. And there's another very important part to this, which I've alluded to before. I did not go into any detail. But if you're looking very carefully at this diagram, these are tanocytes shown here. So they do have cilia. So they're part of those ependymal-like cells. These cells normally have cilia associated with them. And this is why you see on the top all of these little finger-like projections. So they are ciliated. And normally they are kept in close contact with their neighbors through these tight junctional proteins, which you are seeing here. When they start to lose their superoxide dismutase activity, one of the things that they're susceptible to is the production of reactive oxygen species. So when they start not being able to maintain their normal redox reactions and they start to generate these reactive oxygen species, they become compromised. And one of the things that happens is that the tanocyte tight junctions start to break down. So they will produce these reactive oxygen species. In turn, they will produce an local inflammatory response that draws in M1 macrophages, the type of microglia that we looked at in the first lecture series for term test 1. And all of this also leads to the fact that now these are no longer tight junctions. This means that these are much more leaky than they should normally be. So we have an inflammatory response inside the brain. We have a leaky brain now because of the lack of this superoxide dismutase, which is destroying these tanocytes. And as a result, potentially different antigens that might be formed by these macrophages or through other forms of local damage, they may leak out past the normal blood-brain barrier, and they may actually be involved in an immune response as well. So the whole system actually resembles an immune response, but not directly. We're not directly priming the immune response. We're creating a leaky brain, which is what is happening when we damage these tanocytes. And again, the other details, one of the reasons why we like using cuprizone is because it damages many, many areas in the brain, not just one specific area. If I use the other model, so that's model number 2, so EAE is model 1. This is model number 2, cuprizone ingestion. And I went through how cuprizone works in a lot of detail. 2 other models that we can do, and again, they're toxicants. They are toxic to neurons, or they're toxic to dividing cells. So ethidium bromide, for example, you use it in your biolabs, or you used to use it in your biolabs to stain DNA. And it is also toxic to DNA. So it acts to chelate as well as bind between DNA, and it fluoresces as a result. And it can also cause damage to cells. So especially cells that might be dividing, especially cells that are undergoing proliferation, ethidium bromide will act to stop that proliferation. And lysolecithin is a compound that we will inject locally into a small area through stereotactic microinjections, which means that I would have to take the animal, locate the area of the brain that I want to inject into, drill through the skull, take a very small pipette, and then inject very, very small quantities into that area, like the hippocampus, for example, or the corpus callosum, or other areas, to destroy the cells locally with either ethidium bromide or lysolecithin. So that's the third model involving local delivery of these different molecules that act to destroy different types of cells. And this one is the unique advantage of using either lysolecithin or ethidium bromide, is that through very, very precise microdelivery, it will cause focal demyelination. Only a small area will be demyelinated. Cooper's zone demyelinates everything across the brain, and that gives us different views of what the demyelination process might look like. And again, you don't need to worry about all of the details about knowing the differences between these two different inbred strains of mice, the C57 or the BALB-C mice. You don't need to know all the details that are shown to you on this particular slide. So I then transitioned into why this leaky brain is actually really important. And we know from the last lecture that one of the things that we associate with some forms of multiple sclerosis, and again, it's very, very controversial. We don't really know the cause for why multiple sclerosis occurs. Is it vitamin D deficiency? Is it a viral infection? Is it something else? Or this last theory that I'll get to at the very end of today's lecture, is it because you've had concussions when you were in your early teenage years into your 20s? And again, very controversial, but we simply don't know. So one hypothesis is that maybe there's some kind of infection, whether it's a virus or something else that occurs. It causes your brain to become leaky. Some of the antigens that are normally kept behind the blood-brain barrier now get into the bloodstream, and they become target antigens, and potentially lipids and proteins associated with the lipids in myelin might become these target antigens, which will then prime the immune cells that are normally found within the lymph nodes or the lymphoid tissue. So this becomes very much like what we saw earlier, where we are introducing a myelin antigen, and then we are taking out some of these lymphoid tissues and then repriming things. Again, this will then activate a number of different cell types, including the T helper 1 and T helper 17 types of T cells. And then these may in turn migrate back into the brain to cause destruction of myelinating oligodendrocytes and or other types of cells within the brain. And one of the things that we know, if we look at multiple sclerosis tissue, is that around the blood vessels, so what you're looking at here, so this would be a blood vessel, and around the blood vessels, all of these dark points that you are seeing, these represent the lymphocytes that are actually migrating into the brain through those blood vessels. So the blood vessels have become leaky. T lymphocytes can actually move back into the brain, and they may actually induce that damage that we saw a little bit earlier on. Again, this was just to orient you a little bit. You did not need to memorize all of these different types of immune cells that are believed to be involved in the entire cascade of multiple sclerosis, just to let you know that I'm giving you a very simplified version of what we think might happen in multiple sclerosis. We are reasonably confident that there is an immune response, that it is indeed autoimmune. We don't know what triggers that autoimmunity, but we do see a huge increase in the immune response. And ultimately, these are attacking these oligodendrocytes. And whether the cells are the ones that you see here or other cells, that, again, is very controversial. If you look in Science, in the January 25th issue of this year, there was an elegant study that was done to actually help to subtype all of the different immune cells that are in the brain of an EAE animal. And we'll talk about that very briefly at the beginning of the next lecture. But this is just to give you an overview that somehow we are compromising this blood-brain barrier. Somehow these antigens that are normally walled off and not exposed to the immune system are now getting out. And this in turn will cause the migration back of different immune cells that may target a number of cells within the brain. This takes care of the oligodendrocytes, perhaps. It may explain some of the neuronal loss that might be seen, but again, we're not 100% sure of this. The other model that I talked to you about that you also need to make sure that you know, that takes care of the demyelination. But the normal tendency of the brain, wherever possible, as long as you're healthy, is actually a remyelination process. So we haven't really addressed the remyelination. Why does that not occur in multiple sclerosis? And when we look at one of the potential models, there is actually an explanation. So we've opened up the blood-brain barrier somehow, whether it's after a viral infection or something else has happened, or maybe a concussion, and I'll get to that very controversial view in a second. Somehow this blood-brain barrier that is shown to you here is leaky. Antigens can move out, other cell types can move back in, and other molecules that normally do not enter into the brain, things like fibrinogen, which are normally kept inside the capillaries, now is leaky and allows for the movement of fibrinogen into the brain parenchyma, the brain tissue. This fibrinogen, which normally shouldn't be in here, can then act on these oligodendrocyte progenitor cells. And these oligodendrocyte progenitor cells have a receptor for fibrinogen on them. And so the activin 1 receptor type, or ABCR, is the receptor on oligodendrocyte progenitor cells. Normally, we don't have this pathway directly activated. And one of the things that will happen in the presence of fibrinogen is this ACVR receptor is activated. This will then turn on intracellular bone morphogenic protein, or BMP. This in turn will cause the activation of ID2, which is the inhibitor of DNA binding 2, which will then suppress a number of transcription related pathways. The suppression of these pathways blocks the differentiation of these oligodendrocyte progenitor cells into these oligodendrocytes. So this doesn't explain all of the model, but it does explain potentially the problem with remyelination. So if you don't get remyelination, why doesn't it occur? It still goes back to having a leaky blood-brain barrier. So the previous models addressed the loss of oligodendrocytes, so they're being attacked by the immune cells, and you're also not allowing for remyelination by blocking their differentiation. Either way, something has compromised the blood-brain barrier, which is ultimately what we've been looking at throughout the last lecture and a half. So I'm going to switch gears a little bit, but I'm hoping to tie all of this together with the last sort of comments on the paper that I'll be introducing to you. So we haven't really looked at the axon, and we haven't really examined what happens at the level of the axon, other than to tell you that the axon is important. It helps to organize where these voltage-gated sodium channels are. It helps to organize where the myelination is going to occur. They contain different proteins like Casper, contactin, etc. And what happens if we damage the axon? And we know that axonal damage can actually occur in multiple sclerosis, and we talked very briefly about a potential theory about that. What happens when you actually physically damage your brain? So many of you, again, on a very slippery day like it is outside today, you might trip and fall. And is that something that can actually damage your brain? Well, it turns out that having a mild traumatic brain injury or a concussion, and we'll use those terms interchangeably, if you go into clinical practice, you'll know that they're not 100% interchangeable. One of the things that can happen is you can physically damage your brain. Your brain is sitting on your skull. Your skull has ridges, and those ridges can actually tear and rip some of these different axons. In addition to that, just turning your brain in the wrong way through rotational acceleration. So how do you knock someone out? You punch them and you rotate their brain a little bit slower than your skull rotates, and this results in the physical disruption of some of these different axons. You may also cause bleeding in the brain, and this bleeding may result in hemorrhagic foci or small bleeding points. But I also wanted to draw your attention to the fact that the corpus callosum and the dorsal lateral rostral brainstem are two areas, because of where they're sitting and because of the relative size of these connections, are areas that tend to get damaged more often than we think especially in mild traumatic brain injury or where we have rotational injury of the brain. The corpus callosum, what type of tract is that? So this is it's the largest white matter tract that connects the left and right hemispheres of the brain, so it's all white matter tract. And the dorsolateral rostral brainstem area is, again, really important for different things, regulating breathing, et cetera. But we'll talk about the corpus callosum in a little bit more detail because we are talking about myelination. So if you were to go in and examine what happens after traumatic brain injury, whether it's a rotational injury to your brain or you get a blunt force delivered to your brain or you've tripped and fallen, et cetera, there's evidence that there's swelling near the cytoplasm at the site of disruption, and this will eventually cause the loss of some of the cell bodies as well as axonal disruption. It also causes, by the way, degeneration of the white matter tracts. And you have to keep this in mind because typically we concentrate, just like we do for multiple sclerosis, we really only think about the oligodendrocytes, and then we kind of, as a side note, we talk about axonal degeneration, but we also have to think about it this way. White matter tracts also get damaged. White matter tracts are also representing the loss of these oligodendrocytes and potentially other cells like the OPCs. So just keeping that in mind. And again, one of the other things that we know is that as we damage axons, it will also cause the release of intracellular proteins like tau. And tau is normally there to cause microtubule stabilization. And when these axons that are composed of microtubules, they get damaged, tau gets released, and we can start to measure some of the release of tau into the cerebral spinal fluid, et cetera. And this can also cause for their buildup, the buildup of tau inside intact neurons as well. And again, just to orient you a little bit, so when we think of concussion injuries where you've tripped and fallen and then hit your head, your brain actually undergoes damage twice. It's not just a single time. So it's not just when it gets hit. So in red, what you're looking at is the first sort of trauma. And usually what happens is the brain compresses. So as it hits the ground or something hits your head, the brain compresses in that area, but then it rebounds back. So it's like it gets really smaller, and then it elongates again. And this counter-coup is actually really important. It's both the compression as well as the subsequent stretching that is believed to be important in these closed head injuries. And again, a number of things could happen, especially in these highlighted red regions. One of the things that could happen is that you might have neurons actually twisting and the axons being twisted or crushed in the areas that are being under force, or they could actually be torn completely away, and the axons could be completely eliminated. And just again, to keep things in mind, people are looking at MRIs. MRIs by themselves cannot tell you if you have a concussion. There is no reliable indicator right now for a concussion where it's like a mild traumatic brain injury. You know something is wrong with your brain, but there is no physical evidence that something has happened. MRIs cannot tell you that. Diffusion tensor imaging eventually may go that route, but right now there is no diagnostic test that everyone believes that is useful in the diagnosis of concussion or mild traumatic brain injury. So right now, the way that TBI patients are assessed, whether it's locally here at the McIntosh Clinic or if you're going up to Sunnybrook or down to St. Michael's Hospital, you get trauma evaluations, so you get assessed in terms of a physical, et cetera. You will undergo a full neurological exam if you've indicated that your head has been injured. And typically, they will use elements of what is known as the Glasgow Coma Scale. Did you lose consciousness? Do you have motor impairments, cognitive impairments? You get a score out of 15. If you're low on that score, then it's likely that something has happened, et cetera. So again, all of these are sort of checklists that a clinician will put you under. You will get a CT scan of your brain, but that's just to eliminate whether or not there's bleeding that's going on. It does not tell you about swelling, disrupted axons. None of that is evident on an MRI or on a CT scan of your brain. You may, depending on the severity, if they do find bleeding, for example, you may end up having to go to the ICU. And again, depending on what happens, because you will have to know the overall changes in intracranial pressure, the ICP that we'll get to later, you may have to have some surgery, but that's in the very severe cases of TBI where bleeding or mass swelling is actually indicated. So again, for the assessment, most CTs and MRIs are actually normal in concussions or mild traumatic injuries. So there might be functional disturbances, there might be structural injury, so there might be bleeding, there might be something that they can detect as a problem with your skull, but they can't actually tell you, have you sheared your axons? Have you actually induced damage to some of your neurons? None of that is evident on either a CT or on an MRI. So this MRI of a concussed athlete that I got from my colleague, Dr. Manser, looks perfectly normal. There's nothing wrong with the ventricles, nothing wrong with the gray matter, nothing wrong with the white matter, and yet this athlete complained of having a concussion after being hit on the head. And so MRIs are actually not useful diagnostic tools right now. So one of the things that I wanted you to understand is how does the cell damage actually occur? So this gets into mechanoporation, and we tend to think of cytoskeletal proteins as actually being benign and very, very useful structural elements. But one of the things that can happen is that the cytoskeleton, in cases where we have physical trauma, where we are damaging neurons or we have rotational movement, can actually disrupt the cell membrane. And this is the cytoskeleton. Think of it like having your hand in a bag, and suddenly, like, you push your hand through the bag really, really quickly, it'll burst that bag. And when that bursting of that bag or this mechanoporation occurs, this will then break down those gradients that the membrane has been keeping apart. So things like calcium, which is normally high outside and low inside, now there's nothing to stop it from coming inside the cell after mechanoporation. Sodium, water molecules will also traverse into the cell. And again, we tend to think of the cytoskeleton being very, very benign, but it can actually physically disrupt the cell membrane. And by doing so, it can cause a lot of ionic disruption. And again, most of you have that impression because you're growing cells in your labs in a flat medium. And most of the time, you never expose those cells to like shear forces, etc. But they can actually cause a lot of cell death. So again, as a result, you might end up having inflammation. You might have different types of ligand-gated receptors undergoing more activation than they normally would have. We'll talk about this also when we get to stroke later on in the course. And it may also result in oxidative and free radical damage, as well as the entry of things like calcium. Now, this is a really busy slide, and I'm going to be completely honest with you because I want your second term test to be a little bit better. Not that you did poorly, like you, I think, got your midterm multiple choice test marks. We are going to be posting the short answer marks for you soon. There was a transposition error that was one issue where some students that didn't write actually got marks, which was magical, but it was because the marks were inaccurately entered, and we had to figure out like who was supposed to get what mark, and we've had to manually re-enter all the marks from the test papers directly. The other issue, which I'm hoping to address, is that this year we actually only have one returning TA. The two other TAs are new, so they tended to mark a little bit harder than the returning TA. So when you get your short answer marks, I would really encourage you to come in. I will personally remark everyone who asks for a remark, and don't panic. It'll all be okay, I promise. But I do want your second term test to be slightly higher overall. Not that, again, you did poorly. I think we're sort of at the B+ average range, but I was hoping, to be honest, for an A- average. So we are hoping that your next term test will meet that the A- average. So when we look at some of the different forces that lead to this mechanoporation, so some of the cytoskeleton and the cellular strain will lead to this mechanoporation. This means that the axon membrane, as well as the cell membrane at the cell body, they will break. They will have this disruption of the ionic equilibrium. Things like calcium will flow in. It will also cause more intracellular calcium release. This may activate things like caspases, calpanes, more free radicals being generated, etc. And ultimately, as a result of these caspases or other calcium-mediated enzymes like calpanes being activated, it may turn on various pathways, either apoptotic or necrotic, that lead to things like cell death. This in turn may activate inflammatory pathways, but ultimately this means that other factors like calpain will disrupt the cytoskeletal network. Some of that axonal degeneration will be further exacerbated. So this simple act of having your cytoskeleton disrupt your cell membrane or your axonal membrane is enough to initiate this cascade. This cascade is one that you should probably know. Most of it's common sense, but making sure that you know things like calpanes, caspases, why calcium is important after mechanoporation, which might be a new term for some of you. So this idea of mechanoporation of the cell membrane. And again, this gets into diffuse axonal injury. All of this occurs when we have elements of the cytoskeleton being pushed up into either the axolemma, which is the membrane around the axon, or up into the cell body. And again, we don't really think about these glial cells around them, but they are also going to die off as well. And we now know that glial cells will also die after this trauma. And eventually we'll lose some of these different cell types. I think I'm just going to skip through that one for the time being. This is one of the factors that you also need to know for your next term test as well. When you go in, for the most part, if you've just slipped and fallen and you're a healthy individual, you may have a concussion. You may have damage to your brain in certain areas through that coup-counter-coup mechanism. Some of your axons may be damaged. Some of you might have mechanoporation, but in most cases, you may be able to recover. One of the more serious cases is where we have a disruption in what is known as the Monroe-Kelly law. And this is, if you go into neurosurgery, you'll have to know the Monroe-Kelly law. We have to keep the volume of the brain constant. It cannot change. Your brain is encased within the skull. The skull cannot expand. It is a rigid structure. And as a result, this total brain volume has to be continually maintained, which is what is known as the Monroe-Kelly law. Making up parts of this Monroe-Kelly law are, what is the actual volume of the brain itself? It can't expand. So inflammation is bad for the brain because it expands the volume of the brain. In addition, if you have too much CSF or cerebral spinal fluid, that also makes up a portion of the volume of the brain as well. And then there's also the blood flowing through your brain at any given point in time. All three of these factors, if they go beyond the normal expectations, may exceed the normal brain volume, disrupting Monroe-Kelly's law, and then as a result, you'll end up being in trouble. Where does this often occur? This often occurs in places after traumatic brain injury. So one of the things that we know is, not only are we disrupting things at the level of the axon, we are also changing blood flow as a result of the disruption of these axons to certain areas of the brain. So we lose the autoregulation of blood flow to the brain. So that is either going to be completely lost or diminished, or it is a delayed response. So again, what is autoregulation? So when you stand up, your blood pressure automatically adjusts because your blood vessels are autoregulating. That is lost in many individuals after traumatic brain injury, in about 50% of these individuals. That means things like the normal blood flow, the cerebral blood flow in the brain of these individuals after trauma is going to depend on what is known as the cerebral perfusion pressure. And again, you don't need to know all of these details. The details on this slide, yes. All of these are more clinical details. I just wanted you to have an idea of why certain procedures are done. The only other thing to make note of is that this change in cerebral blood flow is going to be decreased in the first 24 hours after mild traumatic brain injury. So we're not talking about where you have like profuse bleeding or we have to remove part of your skull, this is where you've had a severe enough concussion that it's going to impact this cerebral blood flow. And in some studies, there's been a suggestion that this change in cerebral blood flow, this decrease within the first 24 hours, might be an indicator of how long your symptoms of traumatic brain injury will actually last for. And again, it's very hypothetical. We don't know a lot about concussions or TBI. We're still kind of new at this. And in fact, I'm going to show you some data that's only 2 years old that's already being changed or rewritten. So again, there's a lot of effort that's made, especially in very severe trauma cases where one of those indicators that we talked about earlier, that Glasgow coma scale, if you're low on that scale and you've actually lost consciousness, they will monitor a number of different things related to the pressure on your brain. Whether it is an epidural measure or they're measuring the direct pressure inside the brain tissue itself. Again, this slide is not an important one. I wanted to give you a clinical indicator of all of the different ways in which the intracranial pressure has to be measured to make sure that it is maintained. And if it turns out that intracranial pressure is rising and it's too high, in order for this swelling of the brain to be relieved, they will have to remove part of your skull. They'll keep that skull. They'll keep that part of the skull alive so that they can reattach it later. But often it means that there's a little window made so the brain can actually expand into that area. And that's basically to prevent it actually from being pushed down lower into the brain stem region of the skull. And again, there are a number of different mechanisms that clinically people use to reduce that intracranial pressure. And these are very severe cases for the most part. So how do you change intracranial pressure? There are a number of different ways to do this. And again, just to make sure that we're all on the same page, this is a slide that you need to know for your upcoming term test. So how do you what are some of the factors that go into maintaining or reducing this intracranial pressure? One way is to reduce intracranial pressure. You can just remove as much of that cerebral spinal fluid as possible. So remember that earlier diagram about the Monroe-Kelly law? This gets into the cerebral spinal volume, fluid volume, and you can do this in a number of ways. Any ideas what Hob 30 is? You probably have done it if you've ever been in the hospital. Hob 30? Any guesses? Yes, at the back. Not lying down. Okay. So Hob not lying down. That the acronym doesn't quite fit. Can you make the acronym fit? You are right, by the way. Head of bed elevated 30 degrees. So that's one way to actually reduce or drain off the cerebral spinal fluid. And this head of bed 30 degrees means that you're elevating the head of the bed by about this much. So again, you might also have sedation. It might mean that you are diverting some of the cerebral spinal fluid. Another way to do this is to simply reduce the cerebral blood volume. And you can do this in a number of ways, delivering drugs like barbiturates, using mannitol or other forms of hypertonic solutions. All of these different types of mechanisms can be used to reduce that intracranial pressure. And we've looked at the last one. So when it gets too high, it's really to take off part of the skull and to do a small opening in your skull. So again, I just wanted to mention this in passing because it's very controversial. For those of you who have ever had a concussion, one of the things that you're told not to do is to not think. I know it sounds really bizarre, but you're not supposed to engage in cognitive work for a while. And that's really hard for students because you want to get back to school, you want to get back into the classroom, you need to do assignments. And it's also really hard because most of the time nowadays, you are actually looking at screens. What do you know about looking at a screen after concussion or after traumatic brain injury? What happens? Do any of you know? Seizure. No, not that bad. No. So I happen to know this also because my kid has had a couple of concussions because she's fairly active. And again, it's one of those things, if you talk to someone who has had a concussion injury, if they're staring at a screen for a long period of time, they will get headaches. And one of those factors is actually to what is known as the critical flicker frequency. And the frequency at which these monitors that you're looking at now flicker on and off is 60 hertz. For these individuals with traumatic brain injury or concussion, they can actually see that flicker. They may not consciously see that flicker, but they can actually see that flicker depending on the flicker frequency, and it's typically up at around 60 hertz, so they can see that, and that's one of the reasons why they get headaches. It's also one of the reasons why they become very light sensitive. They need that sleep. But surprisingly now, the model system is not where you're resting, not where you're closing your eyes to avoid this critical flicker frequency or CFF. It's now to try to get back into an active state as quickly as possible after a concussion. It's new. It's sort of in the last year or so, but it potentially alters the way that we look at the rehabilitation of the brain, especially after injury. And again, there's nothing on this slide that I wanted you to try to memorize. Just to give you an idea, when you hear about not reading, especially like not looking at computer screens or not watching things on an LCD screen, it's because of this critical flicker frequency that it plays a role in that process. So the last thing that I'm going to get to is this. This has been very, very controversial. So we don't know the cause of multiple sclerosis. And this paper that came out was a prospective study in 2017 that actually suggested very, very controversial. There was a humongous backlash from this particular paper that had come out. And it was very, again, new. The idea was very out there. And you can go look up this particular paper. Plus, I always wanted to introduce a paper by Montgomery Scott, or Scott Montgomery, just a Star Trek reference for those of you wondering. And this is a prospective study of Swedish youth. So they followed these individuals. So they're moving forward in time. So they've had a concussion, then they want to see what happens 5 years, 10 years down the road, and the development of multiple sclerosis, which is what we've been concentrating on. And their idea is kind of interesting. And their idea is that there is a critical period in which these individuals are vulnerable to damage after a concussion. So there's some kind of trauma that occurs, and this is where that concussion comes into play. There is an expansion of this myelin antigen-specific T cell, which is what we've been concentrating on. Somehow it releases different types of cytokines, including things like interferon gamma, and this causes the loss of these oligodendrocytes. And it's been very, very controversial. There has been no follow-up study that actually suggests that this is true, but if you look at some of their prospective data, so this is, again, a prospective study. They track individuals that have had a concussion. So this is from birth to 10 years of age. If they've had no concussions, 1 concussion, or more than 2 concussions during this period of time when they're young, and you look at, do they develop multiple sclerosis down the road, prospectively down the road? And if you look at, for example, the odds ratio, so compared to the normal population, are you at an increased odd? If it's around the number 1, it actually means that there is no difference compared to the general population. And it turns out that if you are getting a concussion, 1 or more than 1, when you were young, you actually have no greater chance or risk of developing multiple sclerosis than anyone else in the population, at least in Sweden. But this result, the next one, is actually the shocking one, the surprising one. That if you are between the ages of 11 and 20 years of age, and you've had 1 concussion or more than 1 concussion, or greater than 2, then your odds ratio actually goes up. It goes up tremendously. So it goes up to more than 1 times. And then if you had more than 2 concussions during this age range of 11 to 20 years of age, you are more than 2 times likely than the rest of the population, 2.33 times the normal population, of developing multiple sclerosis. It has nothing to do with vitamin D, has nothing to do with viral exposure. According to their data, the only thing that results in you developing or having an increased risk of developing multiple sclerosis, where you're 2 times greater than the average population, regardless of sex, etc., is that having multiple concussions when you were younger, in this age range. And again, if you're over that age range, you won't get this. And you might think, okay, maybe it's actually due to your immune system being involved. So I broke a bone while I had this concussion. So broken limbs or bones actually don't contribute to this at all. It's really just concussions. It's been very, very controversial, but that's one of the things that I really want you to think about. If this model is correct, that will change everything that we know about the development of multiple sclerosis. It gives us a different window or a different look into what we should be examining in terms of multiple sclerosis and the immune system. Again, this slide, some of the bullet points on this slide, you probably want to jot down or go back and refresh. I haven't talked to you about interferon gamma so far as an inflammatory cytokine. Sort of unique to this model system that they've incorporated, but I just wanted to draw that to your attention as well. If you're interested, please go back and read this paper. It's not a very hard read. It's very interesting, very controversial. So this is 2019. No studies have actually come out, whether basic research or other clinical studies, either to refute or to support this model. But it is a very interesting and controversial model. So again, just to make sure that we're on the same page for term test 2, on Monday, in Monday's class, I'm actually going to give you one of your short answer questions completely in advance so you can start thinking about that, as well as one of your multiple choice test questions. So it's probably a good idea to make sure that you're there. I want you to, again, let me know if there's anything else I can do to get you ready for term test 2. And I'll see you all on Monday. Be very careful out there. No concussions, no brain injuries.